This procedure allows us to determine patterns of gene expression in zebrafish embryos at high resolution. An antisense riboprobe is hybridized to the complementary target mRNA. The labeled riboprobe is recognized by an antibody that is conjugated to a peroxidase enzyme. A tyramide conjugated fluorophore is added to the embryos, and the peroxidase converts the tyramide to a highly reactive compound, leading to stable deposition of the fluorophore. Unbound tyramide is washed away, revealing the detailed subcellular pattern of mRNA distribution. Expression of a second gene can be observed using a distinctly labeled probe and a different tyramide fluorophore, allowing the detailed comparison of expression.
Hi, I'm Tim Brend from the laboratory of Scott Holley in the Department of Molecular, Cellular and Developmental Biology at Yale University. Today we will show you a procedure for whole mount, high resolution double fluorescent in situ hybridization of zebrafish embryos. In our laboratory we use this procedure to study zebrafish segmentation. So let's get started.
Begin the procedure with embryos that were fixed overnight in the cold in paraformaldehyde. Using a freshly thawed stock of paraformaldehyde for this fixation is critical for successful staining. Remove the fixing solution and wash the embryos at room temperature twice for 5 minutes each time in PBS. Next, remove the chorions of the embryos. The embryos are placed in glass depression plates. Using watch